This video covers Higher Human Biology Unit 3, specifically Key Area 8, Clinical Trials of Vaccines and Drugs. We'll go through the key learning outcomes of explaining why vaccines and drugs are subject to clinical trials before licensing for use, describing how a trial can be made randomized, double-blinded, placebo-controlled, and then explaining why group size and gender balance must be maintained across groups in clinical trials. Vaccines and drugs are subject to clinical trials to establish their safety and efficacy before being licensed for use. There's no point spending millions or billions of pounds making a drug that actually turns out to be no more effective or less effective than a drug that is currently in use. Drugs which are dangerous obviously would be hazardous for health and would make drug companies liable for prosecution. So it's very important to do clinical trials to ensure that a drug is safe before it is licensed for use. The design of clinical trials to test vaccines and drugs uses randomization, it uses double blinding, and it has placebo-controlled protocols also. So subjects in a clinical trial are divided into groups in a randomized way to reduce bias in the distribution of characteristics such as age and gender. This means that the information for, say, 100 participants of a trial will be put into a spreadsheet. The spreadsheet will have their sex, it will have their age, it will have any medical conditions they have, it will have other factors which are relevant to the trial itself. The computer will then arbitrarily and randomly assign people to two or more treatment groups. The experimenter themselves will not know which individuals have been put in which groups and why they've been put in that groups. The computer will make sure that there is a roughly as possible equal number of people in each group of a particular gender, particular medical profile, etc., etc. This means that any results from each group can be compared to each other without people being able to say one group has, for example, more women in it than men, so maybe that's had an effect. If we equalize this across the many groups, then this cannot be part of the bias. Double blinding. In a double-blinded trial, neither the subjects nor the researchers know which group the subjects are in to prevent a biased interpretation of the results. So, for example, if this was a cancer drug, you would be recruiting people into your trial who had potentially very life-threatening and serious cancers. If the researcher formed a positive relationship with one of the participants, they might be more likely to give that person the treatment, so more effective possibly drug than the placebo drug. Because if you knew that person had a previously deadly cancer or other disease, you might like them and want to give them the good drug. But that can't happen because that biases the results. It also means that when the investigator is asking the person about their symptoms, if the person knows they're in the placebo group, they might be more likely to exaggerate the negative symptoms because they know they're not getting the drug. And similarly, a researcher might be more likely to interpret the results. So, for example, if they knew someone was in the group that was being given the trial drug, they would be more likely to have a positive outlook on the person's symptoms. So, to downgrade the serious ones and upgrade the positive ones. So, it's important to double blind. In studies where statistics are being done, which is hopefully most of them, we also treble blind it so that the statistician doesn't know as well. In a placebo-controlled trial, the participants in one of the treatment groups, at the very least, will not be given the trial drug or vaccine. Obviously, in situations, for example, where we're talking about cancer treatments, if there is currently an effective treatment for that condition, you can't just not give the person a drug and make them think they are being given something. So if there is currently an effective treatment for the condition, you, in these trials, would give the person the standard treatment plus the new drug, or the standard treatment plus the placebo. You can't just give them the new drug or just give them placebo in case the drug is harmful or does not have any effect. So again, the placebo would be something that does not contain the trial drug. In a clinical trial, often we're not just looking to see if the drug works, but looking to see what concentration it works in. So maybe there's more than two groups. It might be this group that's getting standard treatment plus placebo, a group who's getting standard treatment plus a low dose of the drug, a group who's getting a standard treatment plus a medium, and a group who are getting standard treatment plus a high dose of the drug. Again, even if you knew you weren't getting, even if you knew you were getting the drug, you wouldn't know what dose you were being giving it in because that's part of the blind test. So that's what placebo is. It's important that group size and gender balance are maintained across groups in a clinical trial. So at the end of the trial, we have to do statistics based on our results. Now, groups which are quite large allow us to reduce what's called the error in our data. So it also allows us to be more confident in our results. So we create something called error bars, which you don't particularly need to know about at higher, and they tell you, so here's our average result for each of the groups. So in this large group, we gave people a drug to see if it affected their test scores in an exam. The placebo group, it looked like got an average result of 10%, so not very good. And the treatment drug group got an average result of 15. Now, because of the range of the actual results people within this group got, and the range here and group sizes, the real result, the actual result for the placebo group is somewhere within this red line. The average might be where the top of the bar is, but the kind of, we're confident the result is somewhere within this red line. And similarly for the treatment group, we're confident the result is somewhere within this line. Now, because there is a gap between the top of this bar and the bottom of that one, it means we're actually fairly confident that there is a difference, that the drug has increased the test score, because even the lowest kind of idea of where the drug effect is, so here, and the highest effect for where the placebo was here, they do not overlap. So there is a bit of a gap. Whereas in a smaller group size, possibly because of the variation in our results and the fact that we weren't able to rule it out, you can see there is overlap. So the top of the placebo group actually goes higher than the bottom of the treatment group, drug group. So we're not completely confident that the placebo isn't actually as effective as the treatment drug, because there is overlap in what we call these error bars. So a larger group size means that you can reduce the magnitude of experimental error, and it means that you can be confident of a statistical difference between the two groups. So we should know that vaccines and drugs are essentially subject to clinical trials to make sure they are effective and safe. You randomise by making sure that the treatment groups are equal in size, if at all possible, and there's an equal spread of people of different genders, different health conditions, different heights, ages, whatever it is that is relevant to the study. And if you block for that, which is the term we use for this, it means that you can essentially then ignore those as factors in your results. Describe how a trial can be double-blinded. So double blinding involves both the participant and the researchers being unaware of which treatment group the participant is in. Describing how a trial can be made placebo-controlled. So a placebo-controlled trial is one where at least one of the treatment groups has not got the active drug under study being given to them. And we've explained why the group size and gender balance has been maintained across groups in a clinical trial. It allows us to reduce the magnitude of our error in the experiment and to be more confident of a statistical difference between the groups. If there's anything you didn't understand of that, please do watch the video again and ask your